Okay, fine. Now, I prepared most common questions in around 50 something pages, but these are, you will not study these questions and then step into the exam. You will study everything. But like, these are the questions that came very frequently in the past years. But this does not mean that you will not study everything. I need to stress on this first. Okay, I chose a question or two on every single topic. Can you hear me?
Yes.
Okay. Now let's start with the lesson classification. What do you remember about dicot plants? What are the characteristics of dicot plants? Quickly. I'm not going to give you the answer. Please, you throw the answer. I'm not going to choose so that we finish the 58 pages.
They have
Branching
two
leaves.
cotyledons.
Okay, I did not hear all of you because you all spoke at the same time, but yes, they have large, broad leaves, if you remember, and they have branching veins, and they have flower parts in multiples of 4 or 5, and they have two cotyledons, if you remember, two primary leaves. So these are the dicot plants, and we will exploit the chance and also revise the monocot. The monocot, they had long leaves or broad leaves. Quickly.
Long leaf.
Long,
Long leaf.
Long leaves and parallel veins,
Parallel
the
veins.
opposite, and flowers in multiples of threes. Yes,
Three.
and the primary leaf, very good. Cotyledons was one
One.
primary leaf. Okay, yes, very good. Now, your syllabus is very fond of fungi. Fungi, it's a kingdom of its own. You need to know its characteristics and its functions and the good uses and the bad uses. State two main features of fungal cells that are used to distinguish them from the cells of the prokaryotes. Quickly.
they
Fungus
have
is
a
a
nucleus
prokaryotic or yes, they have a nucleus, they're eukaryotic. What else?
and wall made of chitin.
Yes, very good. The prokaryotes or bacteria, it's not made up of chitin, it's made up of peptidoglycan. What
The
else?
fungal cells have linear DNA.
Yes, they have linear DNA. Okay. Now, what about the DNA of the prokaryotes? Remind me.
Circular.
Circular.
Circular fungus are eukaryotic, so they have a nucleus, they're larger, they have all the organelles, and the prokaryotes, they're smaller, and they have only ribosomes, and they have circular DNA, and sometimes they have plasmids, sometimes they don't. Okay, so you will, this table that compares between animal, plant cells, fungal, and prokaryotes, and the virus, of course, please don't ignore it. State one morphological feature of this organism that distinguishes it as a crustacean, not an insect. Now let's remember together, what are the characteristics of a crustacean? The number of legs, remind me, I forgot.
5 pairs.
5 pairs.
5 pairs, very good. About the eyes, how many antennae?
1 pair.
Hello, where did you go? I lost connection again.
One pair of antenna.
Okay, are you with me?
Yes.
Yes.
Good. Okay, we disconnect for the 10th time. Is my screen shared?
No.
No.
Yes, we can see it now.
Okay, very good. Now the crustaceans, we said they have 5 pairs of legs. Remind me, how many antennae do they have?
1 pair.
No, crustaceans, they have 2 pairs of antennae. Okay, don't forget that because this is specific for crustaceans. They are like the crabs, the lobsters. Now, insects, how many pairs of legs? This is common. They
3.
like
3.
insects. Yes, 3 pairs of legs. And how many pairs of antennae?
1.
1.
Pair, yeah. And both of them have something in common. What is both? What is common in both of them?
The body segments.
No, they have
Compound
both
eyes.
the compound eyes. Yes.
Compound
Now
eyes.
these, yes, correct. They both have compound eyes. Now these details, sometimes they can leave everything and ask you about those details. Now, which one of them lives on land and which one lives in water and has gills?
Crustaceans live on water and then insects live on land.
Very good. Okay. Now, let's again, another question about prokaryotes, bacteria. I think we already said that. Cell wall made up of peptidoglycan. They're smaller. What is the only organelle that is found in a prokaryote?
Ribosomes.
Ribosomes.
Ribosomes. Okay. And what about the DNA? They're circular or linear?
Circular.
Circular.
Yes, circular. And sometimes they do have a plasmid, sometimes they don't. Okay, now this is done. Now the virus and the viral structure. What is N and what is O? What is N?
Protein coat.
Yes,
Protein
it's
code.
called the protein code. It's called the capsid if you want to know its name. And what is O?
mRNA.
Excuse me? What is O? Did I hear mRNA or I'm wrong?
DNA.
No,
rRNA.
yes. Yes, RNA or DNA. So it is genetic material, either RNA or DNA. If I heard it messenger RNA, this is not in a virus at all. This is in an animal or a plant cell. Okay, now what is this cell? This cell is circular, but it has a cell wall. So what is this? In case they did not tell you.
Fungi.
Yes,
Sorry.
fungi. Yes, and someone said yeast, which is a unicellular fungi. Very good. Now, what is where are genes stored? Choose one quickly. This is so easy.
G.
Gene, yes.
Yeah.
Now let's quickly solve that. What is where aerobic respiration takes place? Which cell structure?
E.
Mitochondria,
E.
which is E. Yes, how do I know? Because it has enfoldings, okay, called crista. And we have here amino acids are assembled to make protein. This is important. Ribosomes. Give me a letter. H. Very good. Okay. Now, describe the similarities and differences between the yeast cell and the structure of a bacterial cell. I think we already answered that. This is really common. Eukaryotes and prokaryotes. Okay? Because the yeast cell is an eukaryote or prokaryote.
Eukaryote.
Eukaryote, of course. So we already said it. Quickly, which is bigger?
Eukaryote.
Okay, which has a nucleus?
Eukaryote.
Which has a circular DNA?
Prokaryote.
Yes, correct. And the eukaryote has linear DNA. Now, you tell me, what are other differences? All the membrane-bound organelles, it has mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, which has ribosomes?
Both.
Both, yes. Very good. I'm trying to confuse you. Which has plasmids, eukaryote or prokaryote?
Prokaryote.
Very good. Okay, next. Now, plants. Now, they love plants. Now, here is a picture that you can discuss without even reading the question. What is happening to this plant and why?
Wilting because of lots of food.
Wilting. Very good. Okay, so they will ask you here to discuss what is happening and use the term tertiary pressure in your answer. And sometimes they tell you use the term water potential in your answer. So we want to use both terms, water potential and tertiary pressure, to explain what is happening here. Can you tell me? What is happening in this plant cell?
Losing
It is
water.
Plant cell is becoming flaccid.
Lost water.
Yes, you all said correct keywords. Someone said flaccid, someone said lost water. Very good. Now, can you explain this again using the term water potential?
The water potential in a cell decrease.
Yes. So inside the plant, the water potential is high or low?
Low.
Low.
Low. Okay. So it is put in a concentrated solution. And the water potential inside the plant, it is low or high? Again, I'll repeat the question because I guess we made a
Low.
mistake.
High.
It's high
It's high compared to outside.
compared to the
Okay.
solution.
Yes. Exactly. So it is put in a concentrated solution with low water pressure outside. And inside the plant, compared to outside, there will be high water pressure. And this will result in what?
Movement of water
Osmosis
by
down
osmosis.
against concentration gradient.
Excellent. You're both correct. Water will move out by osmosis. The keywords, they love the keywords, down a concentration gradient across a semi-permeable membrane. What will happen to the vacuole?
Shrinks.
Shrink.
Shrink.
What will happen to the cell membrane?
It
It
pulls
will
away from the
Detach
cell
from the
wall.
cell wall.
Very good. And it will become flaccid. And how will you put the tertiary pressure in a sentence? The tertiary pressure inside the plant will increase or decrease?
It
decrease.
decreases.
Decreases, lack of support, and that is why it's going to wilt. Now, when I ask you questions, I remind you of things and you give me correct answers. Your problem is that you do not remember all these points alone. So you always give the examiner an incorrect answer. So you can say half of what we said and the rest you just forget it. It's not that you don't know it, you forget it. So please, when you come to solve any essay question, 3 marks or above, squeeze your mind, write everything you remember and do not rush. Do not rush. Write every keyword that crosses your mind, everything related to osmosis and shrinking of vacuole and detachment of cell membrane and tertiary pressure and change in water potential and down concentration gradient. It's full of keywords. Write them all. All. Okay? There is never too much or too little. Just write all the keywords over that point. Explain how mineral ions enter a plant. Very common question. Hmm.
By active
How does ions enter a plant?
transport.
Very good. This is an indirect question. They can ask
Mineral.
you what is active transport. They can ask you compare between active transport and diffusion, but always active transport is part of the question. Here, you will say active transport, but before that, what is the part of the plant that will absorb the ions?
Root.
Root
Yes,
cells.
the root hair cell. The root hair cell gives you a mark. So the root hair cell, it will do active transport, and then you will say everything you know about active transport. Tell me. Now, this is so easy.
Concentration to high concentration.
Very
Against
good.
the
Against the
Against the
concentration
concentration
gradient,
gradient,
needs energy. And don't forget the part that it needs
carrier
carrier
proteins
molecules
change
and the
shape.
very good. You always forget that part, and they like this part. Okay, so it needs energy from respiration or ATP against the concentration gradient, a semi-permeable membrane, and carrier proteins. Carrier proteins. Okay, and this, by the way, all these questions they came from paper 4, and all this is core. We studied this in the core, so don't please ignore the year 9 notes. Now, this picture is very important. We're here a lesson enzymes and human nutrition, etc., etc. They love the villi. Now let's label that because this was one of the hardest labels. Usually the labels are easier. What is P? I put 2 pictures here, a smaller and a very good. This is the lacteal.
Lacteal.
Okay. What is Q?
Goblet cells.
No.
Microvilli. Capillaries.
No.
Epithelial.
Yes. This is not a goblet cell. Okay. Even though goblet cells are present, okay, all along the respiratory tract, but a goblet cell usually it looks like this, and it's usually more common, or you see it in the respiratory system. So this is not a goblet cell. This is, like your friend said, it is an epithelial layer or epithelial cells. The epithelial cells that line the small intestines, and they themselves produce some sort or some types of enzymes. Now, this is not shown in this picture, but those epithelial cells, what do they have?
Microvilli.
Very good. Microvilli. Microvilli, not villi. This is one villi. So, because some of you don't know the difference, this is one villi. And this single villi is lined with epithelial cells. And those cells, they have smaller microvilli, and they both have the same function, which increase the surface area for absorption. Good. What is R?
Capillaries.
Yes, capillaries. Okay. See, if you notice that there is a change in color, this is coming from you And this is going back to S. In case they ask you, but they won't. Even in this question, they didn't. What is U? If they put an arrow and U is towards the capillary, what is U?
Archie.
Arteriole. Okay, you're not far from being correct because the arteriole is a branch from the artery. Correct. So you will be an arteriole that will provide oxygen and nutrients to the villi. And then here we have capillaries that will exchange materials because it itself needs oxygen and nutrients. And what is S? In case they ask you,
Venue.
venue. Very good. Now, we labeled P, Q, R. Now, T. I don't expect you to know what is T, but this lacteal that you told me that P is the lacteal, correct? This lacteal is part of what? Which
Lymphatic
system?
system.
What is
Lymphatic system.
excellent?
Lymphatic.
Very good. So T is the lymphatic system. Okay. It's a little bit too advanced. They will not ask you for details. Even in this question, they said what? Describe the structure of the villus and its role in the alimentary canal. Very easy question. Use the letters in figure 4.2 to support your answer. You don't have to use all the letters. If you use the first three, it is enough, which are the lacteals and the epithelial layer and the capillaries. Okay? But when they tell you use letters, then use letters. Don't ignore this command term. Use the letters. Now tell me, what is the structure of the villus and what is its role and how it is adapted? This is very common. 6 marks.
One cell thick thin walls for shorter
Yes,
distance.
it is one cell thick for faster diffusion. What else?
Large
High blood
surface
supply
area.
to absorb the nutrients.
Large surface area.
Very good. Large surface area. It has microvilli. And they have many capillaries to maintain a concentration gradient and quick absorption. What else? It absorbs nutrients by diffusion, active transport. What does it have so that it enables efficient active transport?
Mitochondria.
Of course, it has everything needed in active transport. Lots of mitochondria to produce energy. It has membrane proteins for active transport. So they will tell you use letters and mention the role of this in the alimentary canal. If you label P, Q, and R, you will take 1 or 2 marks for the labeling. And you will say the lacteal. What is the function of the lacteal? We forgot it.
Absorption of fatty acids.
Absorption of fatty acids and glycerol.
Very good.
Carrying
And
them
don't
away
forget
from
that
the
it
small
will,
intestine.
yes, and sending it back to the blood. What is Q? Or what does Q do? We said what is those epithelial
Contains
cells.
enzymes.
Yes, they do produce enzymes and help in digestion. And they also have microvilli
Absorption.
that does what? Absorption. What is R? Capillaries for what?
Capillaries.
Absorption of glucose.
Yes, excellent. Capillaries will absorb the glucose and the amino acids and the water and everything else. Like the lacteals will absorb the fatty acid and the glycerol and the fat-soluble vitamins and return it to the blood. And the capillaries will absorb everything else. So if you get a long question like this, don't panic. Break it down and you will find that you will have a lot of things to write. I will talk about P and its function, and we just did. Lacteals and what they do. I will talk about Q. I will label it epithelial layer with microvilli, increased surface area. You have a lot of things to say. Absorption, blah, blah, blah. Or you will talk about the capillaries and what do they do. And you will find yourself writing all the information, plus all the adaptations of the small intestine. It is long to increase surface area. It is thin for shorter distance, microvilli to increase surface area, also rich blood supply. It has everything needed for active transport. You'll find yourself having lots of things to say. Okay, so this is an easy question. It's not difficult and it's common. Okay, now enzymes, very easy. What is the product of the breaking down of oil? So easy.
Fatty acids.
Fatty acids and glycerol.
Of course, fatty acid and glycerol. And what is the enzyme that breaks down oil?
Lipase.
Like I should,
Lipase.
we should not even waste time on this. Glycogen. What is glycogen when it is broken down? What does it give you?
Glucose.
The final product, glucose. Yes. Okay. And the maltose. What is the large biological molecule that gives you maltose? No. Maltase is the enzyme. Now, starch. Yes. Yes. Starch is broken down to give you maltose, and the enzyme is amylase. And the organ that that produces this enzyme. What's the organ that
Small
will produce?
intestine.
Very good. Salivary gland produces amylase. Pancreas produces amylase. Intestines produce amylase. Now we know the proteins, of course, protease, amino acids. This is so easy. Explain why lipase cannot be used to catalyze the breakdown of proteins.
Doesn't
Now,
have
this
the
is an
active site to the substrate.
yes, exactly. Because now we have 3 marks. Enzymes are specific. They have a specific shape and a specific active site that has to be complementary to the substrate. Very good. When they bind together, what do they form?
Product.
Enzyme
Enzyme
substrate
substrate
complex.
complex.
Very good. It will not be able to do all that. So this is asking about enzymes, but in an indirect way. Okay? Enzymes are easy and they're very common. Now, the role of bile in the body. I saw this question maybe in so many exams. What is bile doing in the body? Who produces bile?
Modification of fat,
And
neutralize the liver, food that's
Lovely.
coming
You're all
from the
yes, you're all speaking at
stomach.
the same time. You are all correct. Okay. When you come to talk about the role of bile, start from the beginning of the story. Who will produce bile?
The liver.
Liver,
The liver. Who will store it?
The gallbladder.
gallbladder,
The gallbladder. And then it is used in what you all said it, by the way, emulsification of fats, which is physical or chemical.
physical digestion.
Very good.
Physical.
Good. It will now explain. It will break down large fat molecules into
Small soluble.
Smaller
very no, no, no,
soluble
they're not
fat
soluble.
molecules.
No, 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 no. Stop, stop. This is physical. Remove the
Smaller
word
molecules?
insoluble to soluble. Just large to small. Physical digestion breaks large molecules into smaller molecules. That's it. We don't use the terms from insoluble to soluble. We use insoluble to soluble in chemical digestion, not in physical digestion. Okay. There was another question that we answered in the last exam. If you attended the online, this question was a cumulative question talking about all the examples of mechanical digestion in the body. It was a 6-mark question. And we talked about teeth, and we talked about the bile, and we talked about the stomach. If you attended the online, this was a very good question. All 3 of them, the stomach, in case you forgot, the churning action of the stomach, okay, to produce the chyme in the stomach. These 3, they do physical digestion, which is breaking large molecules to smaller molecules only. No chemical change, no insoluble to soluble. Why? What is the objective of physical digestion?
increase surface area,
down the food
Exactly. Into smaller size, only smaller size, no change in chemical digestion. In chemical composition, I stress on that. Why? To increase the surface area to help the enzymes to work next. Exactly. So this bile will break down a large fat droplet into smaller droplets. Only, no change in chemical composition. I will not here change fats into fatty acids and glycerol. No, not yet. Who will do that? The lipase. So please don't get confused. First, we have physical digestion. We'll break down the food into smaller pieces only to increase the surface area so the enzymes can work next. So we are helping the enzymes. Clear? Is this clear? Bile? Yes, ask me.
In this question, about the physical digestion, can I mention peristalsis of the esophagus?
No.
I saw
Why?
it in one of
Because
the marking schemes before.
maybe it's not wrong, but peristalsis is not going to do digestion. It moves like it's in the esophagus to push the food. Okay, it's in the ureter to push the urine out of the down the ureter out of the body. It usually pushes the food, and also the movement of the alimentary canal is a long muscular tube. This long muscular tube from the mouth to the anus, it contracts and relaxes. These contractions and relaxations are called peristalsis. So it's not wrong. Okay, it depends where are you along the digestive tract. If you are in the esophagus, you're doing peristalsis, but is this peristalsis helping in digestion? Tell me.
No, it's moving the food.
Exactly. So it depends on where are you in the digestive system. If you are in the stomach, then yes, it is digesting because we have contractions and relaxations. It's called peristalsis, but in the stomach, we have a specific name called churning action. So the churning, you use it with the stomach. Peristalsis, you use it with the esophagus to push the food. Okay? Clear?
Okay.
So it depends on where you are. All these terms mean the same thing because The digestive system is a tube of muscle, and any muscle will contract and relax. They're just different names, but they all mean the same thing. Okay, I hope the difference between physical and chemical is clear. Now, this graph is very important. We talked about it 101 times, which is the effect of pH on enzyme activity. And the second one here, the effect of temperature on enzyme activity. This question is very common, more than common. Now, here you have the effect of pH on enzyme activity. And then they will ask you, describe and explain the effect of pH on the activity of sucrase shown in the figure. How many marks? 6 marks. Now you will go to the figure without even going to the figure. What is the effect of pH on the activity of enzymes? What are the 6 marks? Where do they go?
pH causes it to denature.
Okay, this is the only thing you will say when you have a question out of 6 marks?
No, the
And
first part,
mentioned
which
the
where
optimum pH.
the enzyme
The optimum
activity is increasing, and then the optimum, and then when it starts to when the pH starts to increase above the optimum.
Yes. Okay. 2 were speaking here, and you're both correct. I heard you both. In the pH graph, you can either start from here or you can start from the very beginning. Like Iman did, she said, from the very beginning, you will divide the graph into 3 parts. As the pH is increasing, the percentage activity of the sucrase is increasing until it reaches the optimum. Now, when you are talking about this part of the graph and you're telling me that the percentage activity of the sucrase or the enzyme is increasing, what is increasing exactly here? Here. What do you say here in this part of the graph?
Enzyme
Enzyme
activity.
substrate
substrate
complex.
Yes, you are right. There is more successful collisions, more enzyme substrate complex. These 2 sentences will give you a mark. And then what do you say here?
Optimum pH.
It reaches
Enzyme
Yes,
optimum
optimum
activity.
means maximum. Just change the term. Maximum rate of reaction, maximum successful collisions, maximum enzyme substrate complex. Okay. Now, also, if you want to specifically talk about this graph and compare this line with this line, which line is steeper and showing a faster increase
Line
in
B.
percentage activity? B. Yes. Okay. So this is the steeper part of the graph. So we all agree that as the pH is increasing and moving close to the optimum, the activity of the enzyme is increasing, and it will increase very quickly in the second part of the graph from pH 5 to pH close to 7. Okay? Because we have more successful collisions, more enzyme substrate complex, etc., etc. Here, optimum. The word optimum pH is very important. Maximum rate, maximum successful collisions, maximum enzyme substrate complex, maximum production of product, whatever. And then here, what do we say? We're moving away from the optimum. What is happening?
Enzymes denature. No more
Yes. And
sucrase.
very good. The active site does not fit into the substrate, not complementary anymore, no successful collisions, exactly the opposite. Now, do we mention anything related to kinetic energy in this
No.
graph? Never, okay? Now, if the pH is very far away from the optimum, like if the pH is 2 and it remains at 2, what will also happen to the enzyme?
denature.
Yes, it does. Okay. So we start, when we talk about this type of graph, we are saying that the pH is moving towards the optimum. That is why the percentage activity will increase until it reaches maximum when we reach the optimum pH. But if the pH is far away from the optimum at the extreme ends, whether at 2 or at 12, and the optimum is near 7, it will also denature here and denature here. Okay. So you will start by saying, as the pH is moving towards the optimum, the percentage activity of the sucrase enzyme is increasing because I have more successful collisions and more enzyme substrate complex and more and more and more. Here, optimum or maximum, everything is maximum. Here, you discuss denature in detail. Change in active site, no longer fit, no enzyme substrate complex, it will no longer bind with the enzyme, etc. And if the pH is far away from the optimum and it remains this way for enough time, the enzyme will denature, whether at 2 or at 12. Is this clear?
Yes.
I hope it is. Now, I'll send you the mark scheme, by the way, when we're done.
Doctor,
Now,
what
temperature.
is the
Yes,
difference
my dear.
between gradually and steeply?
Gradually, this is a very nice question. I'll tell you. Gradually, let's compare between two lines. You have two lines moving upwards. Okay, which one is moving faster, A or B?
A.
Let's make it more clear. Yes, let's say A is like this. See, A is moving, is much steeper. Steeper means it's more straighter. It's moving, they're both increasing, but B is slow or gradual, and A is steeper or faster. Okay? If they're both decreasing, which one is decreasing faster? A or B?
A, the one that's deeper.
Yes. Yes. Okay. It's decreasing faster. Like they're both, one can be decreasing like this and one can be decreasing like this, dropping down. So A will be steeper than B. A will be decreasing faster than B. Okay. Good question. Now, the temperature. Exactly the same shape, but we say different things. When we have low temperature, what does this do to the enzyme activity?
Low activity.
Yes. It's fast or slow enzyme action?
Slow because it has little kinetic energy, less collisions per unit time.
Very good. Less enzyme substrate complex. Never here will we say it will denature. No, it only denatures at very high temperatures. Here it's cold. The temperature is very cold. So I have little kinetic energy. So we do exactly, or we say exactly the same thing, but we mention kinetic energy with temperature and we do not mention kinetic energy with pH. So here at low temperature, this is cold. Enzymes are slow, little kinetic energy, little collisions, little enzyme substrate complex, and then it will increase here, more kinetic energy. They're moving faster. Just add this. Here, optimum temperature also, maximum everything. And here, the same, it will denature. Very easy questions. I hope you get questions like these because you're very good at answering those questions. Okay. Now, just in case you get another graph that looks like that, this shape, how do we describe it on a graph? Up and down, up and down, up and down. Excuse me?
Fluctuates.
Yes, it fluctuates. Very good. And if it's straight like this,
Constant.
constant levels of plateaus, whatever. Now, here is a tissue inside a plant. Okay, which tissue is that? They tell you it transports water and
Xylem.
ions. Xylem. They love the xylem. So this is the xylem. Describe how is it adapted. Very easy and a common question. How is the xylem adapted?
Hollow.
No
It's hollow.
place.
What else? I don't like short answers. Hollow. Huh? What
No
else?
organelles, no
Walls.
nucleus. Or it is
Exactly.
a
No
feature of water
nucleus. It
It has
has.
lignin.
lignin in its cell wall.
Yes, you're all correct. You're all amazing. And don't forget the degenerated end wall so that water will flow without resistance in one-way direction so that it doesn't
Lignin to
collapse.
support. For support.
Very good. Yes, you're all right. Now, carbon dioxide. Now, this is so easy. Carbon dioxide gases, how do they move anywhere?
Diffusion.
Diffusion.
Diffusion, of course. Now, this question is nice. Describe the pathway taken by a molecule of carbon dioxide from the air outside a leaf into a spongy mesophyll cell.
Enters the leaf through the stomata by diffusion down a
Very
concentration
good.
gradient.
Yes,
Then
yes,
moves to the air spaces. Diffuses in air spaces.
yes, yes, very good. Here, the CO2 is moving in the opposite direction, not from in to out, but out to in. You will mention somata controlled by guard cells, and then the air spaces, it will mirror the air spaces first, and then it will enter the mesophyll cells. This is core. Okay. State the term given to something present in the environment in such short supply that it will restrict life
factor.
Limiting
processes.
factor.
Yes,
Limiting
they love
factor.
limiting factor. They love it. They love limiting factor. Limiting factors in plants that are always tested are usually temperature, carbon dioxide concentration, and light intensity. Those are the three factors that they can ask you about, and especially in paper 6. If the limiting factor here is plotted on the x-axis and the graph moves in this direction, what is the limiting factor at A if light is being tested here?
Light.
Light.
Light. Okay. And then at B, it is any other factor other than light, because we are increasing light intensity. And even though we're doing that, the rate of photosynthesis remains the same. Good. State one other feature of environment that may also restrict the rate of photosynthesis. We
Light
just
intensity.
said them. Very good. Don't say
You
water, because usually water is available. It's not wrong,
you
but
made
water is usually in enough supply. Okay. Very good. CO2 concentrations. Now, this is important. This was a November 22 exam. Where is the xylem? Because in this part of the question, they ask you where the xylem. Let me, A or B?
A.
Yes, A is the xylem. A is, or the xylem in a stem is always located inside or inwards. Okay. And the phloem is B. Okay. Now, what is this? In case they ask you, but I doubt, what is this part of the
upper
whatever?
Epidermis.
Yes, it is the epidermis, correct. Mhm. Who has questions?
Miss, the
Anyone has
xylem in the
questions?
stem, it's closer to the end walls, right?
Miss.
No, my dear. Look, you have three parts of the plant. You have the root, and the xylem is in the center of the center. Like they're both in the center, the xylem and the phloem. The vascular bundle is in the center, and the xylem is in the center.
The inward,
Okay?
yeah.
Yes. And you have the stem, like this one. And the vascular bundle is located in a ring. Look at the pictures. And the xylem is also towards the center of the ring. This is the xylem. Okay, my dear?
Okay.
The part that you were confused at is the leaf. This is the leaf. And you have a vascular bundle in the leaf. And the xylem is towards the upper surface of the leaf. This is the part that confuses you.
Okay.
This is the
Thank you,
leaf.
Miss.
Okay? You're welcome, my dear. Go back and look at the pictures. Now,
Okay.
we always talk about the xylem and we forget the phloem. What is the function of the phloem?
Translocation.
Yes, the word translocation is very important. It will translocate. What does it translocate?
Sucrose and amino acids.
Very good. From where to where?
Source to sink.
Source from the
Excellent.
root hair
Can you
stem,
someone remind me, please, what is the source?
where it's produced.
Where it's
Producing.
produced.
Zero.
Exactly.
Sink is where
And
Sink
when
is
we
where it's
have,
stored or used.
yes, okay. Now, to avoid confusion, sometimes they use young leaf and adult leaf. I don't know if you noticed that. If you have an adult leaf, this means that this leaf is doing photosynthesis. So this leaf, if it is an adult leaf, then it will definitely be the source. If they are telling you or they use the term young leaf or growing plant parts or growing leaves, it's young, it's a baby. This leaf, is it photosynthesizing or not yet?
Not yet.
Not yet.
Very good. So this leaf will be a source or a sink?
A sink.
sink.
Yes, yes. Don't make them confuse you because they like to play around with words. Adult leaf, this means it's photosynthesizing. Young leaf or growing parts of plants means it's growing. It needs energy. It needs sucrose and amino acids. So it will be a sink. So the sink can be a storage organ, a root, growing leaf, growing plant part. Okay. And it changes according to the seasons of the year. Now, transport in animals. Let's label. What is C?
Aorta.
Aorta.
Yes, the C is the aorta. Please be careful of the spelling. Now, what is A?
Pulmonary artery.
Good. What is this arrow in case they label you? What is going inside here?
Pulmonary vein.
Very good. That's B, yeah. And then they told you this is the septum. What is C?
Septum.
Vena cava.
No, sorry.
It
Vena
is
cava.
no. Look, you got confused. We just labeled it. C is the aorta. This is just
Aorta.
another branch behind the heart. Okay. Now, what is D?
Vena cava.
Inferior vena cava.
Vena cava.
Very good. Inferior vena cava. And what is this arrow towards the heart,
Superior
which we already labeled? This is the superior
pulmonary
vena cava.
vein.
Look at the arrow. Yes, pulmonary vein. Now they love double circulation and single circulation, if you noticed. What are the advantages of a double circulation?
Separates
In the
oxygenated, deoxygenated blood.
in systemic circulation, blood is of high blood pressure. So faster, tissues receive oxygen, glucose faster, and it takes away carbon dioxide and waste products faster as well.
Yes. Very good. Perfect. One of you said it separates oxygenated from deoxygenated blood. Very good. And then another one said that you have systemic and pulmonary circulations. Now, this has to be mentioned. Every time you talk about a double circulation, define it. Start by defining it, because when you define it, you will say that there are two circulations, one systemic, one pulmonary. You will talk about the characteristics of this, and you will talk about the characteristics of this. Which one has high pressure circulation?
Systemic circulation.
Very good. And just like your friend said, this is important to deliver the oxygen and the nutrients quickly and to also remove the waste products quickly, such as the CO2. And the pulmonary circulation, high pressure or low pressure?
Low pressure to prevent damage and rupture of the delicate
avoid
alveoli.
Yes, thank you very much. Okay. Then, if you compare that, I know that they're not asking you this in this question, but it's very common. If you can compare this circulation with the circulation of fish, which one is efficient?
The
Double
double.
The double circulation.
Yes, very good. And don't forget to say that also when we have a high pressure systemic circulation and we are sending blood quickly, we're not only sending glucose and oxygen, we're also providing heat. And this is very important in
Or warm-blooded animals.
warm-blooded animals. Yes. Fish, their circulation is described as single circulation and it is inefficient because the blood is traveling through the body of the fish slowly. That is why the fish has to be moving the whole
Moving
time, even
constantly.
while it's asleep. Very good. Okay. Now let's label that. Let me see. I want everyone to join. Like I have, what is, we already labeled everything related to the heart. What is C? C is coming out of the small intestine and going to the liver.
The
Hepatic portal vein.
renal vein.
Excellent. Hepatic portal vein. Now, we did say that the vein or the artery coming out of the lungs going to the heart. Tell me, what is it?
Pulmonary.
Yes,
Pulmonary vein.
what? Pulmonary vein, correct. Now coming out of the heart, going to the lungs.
Pulmonary artery.
Pulmonary artery. What is F? Going towards the heart.
Vena cava.
Vena cava. Okay. So we already labeled that. State the letter from figure 1.1 that represents the blood vessel that contains blood with the highest concentration of oxygen. Which one has the highest oxygen?
The pulmonary vein.
Pulmonary.
B.
Yes,
vein.
it's B. Correct. It is B because it's directly coming out of the lungs. Okay. State the letter from the figure that represents the hepatic portal vein. We already labeled that. That was the first thing we labeled, which is what?
C.
C. Okay. State all the letters that represent arteries. Anything going towards the heart or away from the heart?
Away from the heart.
Away from the heart. Yes. So you will look for the arrows going out of the heart, which is A and D and E. Correct. Okay. Explain the importance of the part labeled Y. Now, this is also very common. What is Y?
The septum.
Septum. Why is it important?
Allows the blood, divides the heart into two sides.
Separates the oxygen and oxygenated blood.
Yes, all of you are correct. You can give only two advantages. It divides the heart into the artery and the ventricles, oxygenated and deoxygenated, provides for a double circulation. Good. Now,
Can I say
this,
different pressure?
excuse me?
Can I say different pressure?
No, because it divides the heart. What do you mean by different pressure?
I mean إن هو ال left side بيبقى high pressure عشان provide البودي كله with blood faster يعني وكده.
It separates the left side of the heart from the right side of the heart. It is vertical. Okay? When you are talking about the atria and the ventricles, then if it was horizontal, maybe what you will say is right. Okay, let me see. Explain the importance of the part labeled Y. What is this exam? This is March 23. Let's look. Where am I? We said March 23. There's only one paper 4. Which question was that? Do you remember? Let us look. This is 1 IV. 1 what? 1A IV. Let's see if this was in the mark scheme, but I don't think so. 1A. No, this is not the correct one. This is March 23, we said?
Yes, 2023, 42, February, March.
Miss, here it is.
1
1A.
here. See? Separate the left and the right sides of the heart. We said that separates the right atria and ventricle from the left atria and left ventricle. Keep deoxygenated blood and oxygenated blood separate. It did not mention anything about pressure. So don't mention anything about pressure. Okay? Most probably, yeah. It separates the left and the right sides. It provides for a double circulation. It does not allow the mixing of the oxygenated and the deoxygenated, like we know. Okay, but it doesn't mention anything about pressure. So don't add that. Okay? Okay. Fine. Now, where are we? Okay, here we have arteries and it will divide into smaller. What is X?
Arterioles.
Arterioles. Now, explain reasons for the changes in pressure seen in the arteries. The arteries, as you can see, we said the pressure is fluctuating. Why? I was expecting also you to get confused in this question. Arteries, what are the walls of the arteries made up of?
Elastic tissue
Yeah, and
muscles.
Fiber.
very
And
good.
what
Very
happens by high pressure? It expands and then gets thin again.
Yes, arteries and veins, these are tubes made up of muscle. They have smooth muscle in their walls. We did study their structure. Arteries, they have a narrow lumen, and the veins, they have a wide lumen, if you remember. And
Yes.
all this is muscular wall and elastic tissue. And this is muscular wall and elastic tissue. As long as we have any sort of muscle, this means it will what and what?
It allows for elasticity and
Contract
shape,
or relax.
right?
Very good. It will contract and relax, and the elastic tissue will make it elastic, just like you said, and explain the reasons for the change in pressure in the arteries. Now, this artery, let's think of the aorta. This is your heart, and this is the aorta. The aorta is just above the what? What is down there that is contracting?
The left ventricle.
Very good. So when the left ventricle contracts, there will be high pressure in the artery. And when the left ventricle relaxes, there will be low pressure in the artery. Okay? So the high and the low pressure in the artery is the result of the high and the low pressure in the heart. While the heart is pumping, the heart will pump, it will contract. And then the pressure will be high in the arteries. And then the heart will relax. There will be low pressure in the arteries. Okay, these arteries themselves are made up of muscles and elastic tissue to be able to withstand the changes in pressure, to be able to withstand the high pressure and the low pressure. Okay, did you understand this question? This was a tricky question. We know that the heart is made up from muscle, and we have very thick muscles in the ventricles because the ventricles, they contract really strong. When the ventricles contract, when the heart contracts in general, then and the blood moves from the ventricles directly into the arteries. So it is taking all this high pressure from the left ventricle and into the arteries. So the arteries will be exposed to high pressure while the heart is contracting, and it will be exposed to low pressure while the heart is relaxing. That is why the arteries, they have to have thick walls because they carry blood at high or low pressure.
High pressure.
Hi.
High pressure. Okay, so there are pressure changes because the heart is contracting and relaxing. When it is contracting, high pressure in the artery. When it is relaxing, low pressure in the arteries. Okay, and we're specifically talking about the ventricles. And these arteries, they have to be made up of thick walls to withstand the pressure. Good, due to the contraction of the muscles in the heart. Now, using the information, explain the structural adaptations of arteries and veins. You have to here remember arteries, how do they look like? And veins, how do they look like? Now, I will not tell you anything.
Arteries
Remind me.
have narrow lumen.
Yes, very good.
Arteries are
What,
made up
what
of
about
thick
veins?
muscular walls
Valves,
Yes.
veins have
with
walls.
elastic tissue.
And veins also, by the way, have muscular walls and elastic tissue. But what's
But
the
it is
difference?
thinner. The veins are
Yes,
thinner
it's thinner. Okay. Yes, very good. Why thin?
because the arteries
Arteries
carry
are
blood at a
perfect.
high pressure.
Yes, Iman, my dear, you're amazing. You're perfect. You're outstanding. Just give a chance for others to speak, my dear. Okay.
Okay.
So, someone else was speaking. Can you tell me why veins are thinner? Hello. Why veins
Because
are
it
thinner? They
carries
carry.
blood with less blood pressure than the artery.
Very good. Very good. This is low pressure. This is high pressure, which has to overcome more resistance, which has a pulse. We have a nice table.
Artery,
Exactly. The artery has, talk about the lumen. What is the lumen here in the artery?
Now,
narrow,
Narrow lumen, and this has a wide lumen because, again, because this will carry
veins,
blood at low pressure. Who has valves,
veins
the
carry
arteries or the veins?
veins,
veins,
Okay. You have a nice table. I
and
don't know where in
veins
10 or 9.
also
Huh?
oxygenated blood.
Exactly. Prevents the backflow of blood. So you
Miss,
have a lot of things to say. Yes, my dear.
can I say it has a narrow lumen to withstand the high pressure
Yes. To maintain high pressure. Because
to
the narrower
maintain
it
it?
is, yes, yes,
Okay.
yes, yes. You have a table comparing the structure and pictures. I don't remember it's year 9 or 10, but we have a nice table somewhere.
It's
Okay,
there in both.
very good. Okay, explain why people now. This is which lesson? Disease and immunity. This is a very important lesson. Immunity. Explain why people were not protected from cholera immediately after receiving the vaccine.
No memory cells were created.
No memory cells were created. Now, why did I choose this question? Because it's a very tricky question. This question can be
The
changed.
cholera has not
Takes
reached
time.
the small intestine yet.
No, 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 no,
Miss,
no.
it takes
Look,
time. It's a lot. It's a time-taking
exactly,
process.
exactly. Now, this question can be a direct question, but they don't like to give you direct questions. They like to confuse you. They can ask you what happens in your body when the cholera bacteria enters. Okay? And then the answer will be the same because when any foreign body enters your body, tell me what happens when the bacteria
the
enters.
antigen positive,
Immune system is
The
activated.
immunity is active. The immune system is
Yeah.
activated.
Yes, very good.
It
and
After the
selects
immune
the
system,
Antibodies.
selects complementary
very good.
lymphocytes
Yes, the lymphocytes will produce antibodies.
produced
You're
by
all correct.
mitosis.
mitosis,
Antibodies, yes, the lymphocytes, the chosen one, will divide by mitosis to
to
increase in number and produce antibodies, specific, don't forget, specific antibodies. And then these antibodies will kill the cholera. And after all this is finished, the war is finished, some lymphocytes remain in the blood and they become
Memory cells.
Memory cells.
memory cells. Very good. And when this happens, you will become immune. Yes? So if you get or infected with cholera again, will you be sick?
No, it takes a shorter
No.
time.
Yes, very good. So this will give you lifelong immunity. But is it immediate protection or this protection is not immediate?
Not immediate.
Not immediate.
Not immediate. Now let's go back to the question. Explain why people were not protected from cholera immediately after receiving the vaccine.
takes time to mitosis and find
Exactly. The answer that we said together is exactly the same. This vaccine contains the weak pathogen. And when the weak pathogen enters the body, it's like an antigen. It will activate the immune system. We will choose the lymphocyte that will produce the complementary antibody. The chosen lymphocyte will divide by mitosis. The antibody will do the job. And then some lymphocytes will remain and they will become memory cells. So this will take time. That is why people that were not protected from cholera immediately, because it takes time. You understand? So this is such a twisted question. And the answer is the same. If the question was direct, what happens in the body when you are infected with cholera? You will find that the question is nice and easy and you will be happy and satisfied. But when they get you this question, you will panic. Explain why the people were not protected from the cholera immediately after receiving the vaccine. How will you answer the question? You will mention what happens to your body when you take the vaccine, which is the same answer to what happens to your body when there is a foreign antigen. You will explain all the steps and you will simply write that this takes time. This will not be immediate acting. It has an advantage. It will provide you with long-term immunity, but it takes time. Remember when I told you, change the title in your notes, and the title was what happens to your body when an antigen enters, and the same answer you will write what happens in your body when you take a vaccine, because the vaccine is an antigen, but it's just an artificial one. It's just a weaker one. Are you all with me?
Yes.
So please, the vaccine is very important. It's really, really common. What happens when you take a vaccine is exactly the same answer to what happens when you are infected with a pathogen, because the vaccine is a pathogen, but it's a weaker one. So if you take a vaccine, we give a particular answer. And when you are infected with the real pathogen, you give the same answer. Your body will do the same thing. Your immune system will become activated and so on. Okay, so I hope questions like these don't panic you. Just try to think, what do they want from you? Doctors ensured that people who received the new vaccine had not had cholera before. No, we will not be able to answer this question because it's part of a question that it's not there. I took part of it. Okay, explain how cholera causes diarrhea. Now, this is also very common.
The bacteria produces toxins.
Yes, which is the first thing that you have to say.
Attached to the intestinal wall.
Stimulates
Yes.
hydrochloric acid.
Water gets out by osmosis.
Yes, the intestinal walls, and then when it is irritated, the chloride ions will move out from where to where?
From the lumen.
Small intestine to the gut.
From the capillaries and the small intestine into the lumen. So at the inside of the small intestine or the lumen here, or the gut, they call it the gut, will become very concentrated with salts. And then what?
Dehydration.
Before that, don't jump. What will follow the salts? You have here the
Water.
small
The
intestine. Yes,
osmosis. Water moves by osmosis down a concentration gradient.
excellent. Very good. And then you will have here inside the small intestine lots of water and lots of salts and digested food. All this will not be absorbed, and it will move very quickly along the small intestine, and it will come out of your anus as watery feces, and this is diarrhea. Don't forget that. This question is really common. Okay? And when you lose lots of water, this is when you will suffer from dehydration. Okay, so please memorize the sequence of events. This question was common. Always start with the release of toxins by the bacteria. They will irritate the intestinal walls. The intestinal walls will release salts, chloride salts. The inside of the lumen will become very concentrated. This is going to attract water from the surrounding capillaries by osmosis. The inside of the lumen will be filled with water and salts, and all this will go out as watery feces. You will lose lots of water. You will be dehydrated. Okay. This question, 22 exam, outline the consequences of the changes in the number of lymphocytes for the health of the person infected with HIV. Now, I like to choose difficult questions or questions that are not direct. What do you know about HIV?
It destroys the
Virus?
lymphocytes.
It what?
It destroys lymphocytes.
Yes, very good. And when the HIV destroys your lymphocytes, what happens to your immune system?
You're no longer
It
immune.
becomes weak.
Very good. But it's not enough to say it will become weak. When you have no lymphocytes, what will not be produced?
Antibodies.
No antibodies.
Exactly. No antibodies, no memory
No
cells.
memory cells.
Very good. No lifelong immunity, very weak or poor immunity. And when you have very poor immunity,
You become
what?
diseased.
Yes, you will become very sick for any reason. Like you will have many infections. They are called opportunistic infections, if you remember. Okay. And you can die from something as simple as the flu. Okay. So, And we also don't forget that HIV is bloodborne, transmitted through contaminated blood products or from a pregnant mother, sexually transmitted disease. Explain why antibiotics are not used to treat viral infections. An easy question, but very common.
Because virus are non-living
No
and they
cell
don't
wall.
have any cell wall.
Yes, the virus is not living anyway, and it doesn't have any cell structures or cell wall or whatever. Okay, respiration. They always ask this question. Physical activity and how it affects the body. Physical activity changes concentration of carbon dioxide. State where this change is detected and how the body responds. This question can be out of 2 marks or 4 marks. That same question. Who will detect?
The brain,
Uh,
high carbon.
exactly. Exactly. High carbon dioxide will be detected by the brain. And then what? What will the brain do?
Send the
To
impulses to the
increase the
motor
rate and
neuron.
depth of breathing.
Very good. The brain will send impulses. Now, when the impulses move from the brain to the lung, this is which type of impulse? Motor or sensory?
Motor.
Motor.
Very good. Yes, I'm just asking you a little of details. So the brain will send an impulse to the lungs, to the muscles in the lungs. Which muscles exactly?
Cardiac.
Excuse me? What are the muscles in the lungs that we studied?
الinternal
Internal and intercostal and
intercostal
Yes,
intercostal.
the intercostal muscles and what else? The one at the base that
the
is curved.
diaphragm.
Yes, the diaphragm and the external intercostal muscles, because we will increase the rate and the depth of breathing. Okay? We will start by increasing. And why do we increase the rate and depth of breathing? Let's just dive in case they ask
to
you, explain reasons.
retrieve the oxygen lost.
Okay,
Repay oxygen depth.
to repay the oxygen depth and to remove the excess what?
Carbon dioxide.
Yes. Look, sometimes they ask you, why does the rate and the depth of breathing increase during the exercise and after the exercise? And these are two different answers. You have them also as a table in this lesson. During exercise, your breathing rate will increase, of course, and depth. Why?
Or oxygen for aerobic respiration to produce energy.
Very good, because
Get
I
rid
will be
of
providing, excuse me?
do you hear say get rid of lactic acid?
No, we say it if we are asked after exercise. This is the point I wanted to specify. If you are exercising, your rate and your depth of breathing will increase during the exercise. Why? To provide oxygen for aerobic respiration, to produce energy because your muscles want to contract, and also to remove excess what? CO2. Okay, so it does not build up inside the body because CO2 makes your blood acidic, and this is a big problem. Now, why does your breathing rate keep on increasing after exercise? The exercise is finished, but you're still breathing heavily. Here, you mentioned the oxygen depth. Okay, I need oxygen after the exercise is finished because I need to repay the oxygen depth because this oxygen will be used to do what?
Oxidize lactic acid in the liver.
Very good. To oxidize the lactic acid in the liver. This lactic acid will be carried by the blood. It will go to the liver. And in the liver, we will need the oxygen that you will breathe in after the exercise to oxidize the lactic acid back to carbon dioxide and water. Okay? So this part is very common. I'm exercising, I will breathe heavily. Even after the exercise, I will breathe heavily. During my exercise, I will give an answer. After my exercise, I will breathe heavily. I will give another answer. During my exercise, I will need oxygen for these reasons, and I will remove the CO2 that is building up in my body. After the exercise, why am I still breathing? The exercise is finished. Here, I will mention to repay the oxygen depth, and you mention the whole fluid. Okay? And if you do mention oxygen depth during, it's not wrong, but you need to understand the difference, especially after exercise. Why do you breathe heavily? Don't say I need oxygen because the muscles are contracting. No, they're not contracting anymore. The exercise is finished. I need here to repay the oxygen debt. Is this part clear? Hello? Is it clear?
Yes.
Okay. Now the percentages. You need to memorize those numbers. Carbon dioxide in inspired air. What is the percentage?
0.04.
Very good. And outside expired air. When you exhale, more carbon dioxide or less? More.
More.
So inside 0.04 and outside 4. It's easy to remember. They're all fours. Oxygen in inside air. How much goes in?
21.
20 to 21. And how much goes out?
16.
16. Okay, now the alveoli and the capillary. Again, how is the capillary related to the function, to its function, the capillary? And then we will talk about the alveoli. I think we said that or we didn't know. We talked about arteries and veins. Now, how is the capillary related to the function?
One cell thick.
They're
cell
only
thick
one
with
cell thick for
Exactly.
shorter diffusion distance.
Yes, what else?
Have pores for
Porous.
heat.
Yes, it has pores. What else? What about the lumen of the capillary? It's wide or very, very, very narrow?
Narrow.
Very narrow, so that blood travels quickly or slowly?
Slowly.
Slowly to give time for diffusion. Go back and read it. What else? We said it's porous, it's one cell thick, and also we have many capillaries or little capillaries?
Many
Many
capillaries.
capillaries for
Many
larger surface
capillaries.
area.
Yes, and exchange of materials. Okay. Now, where is that? I don't know. But what are the adaptations of the alveoli in case they ask you for the adaptations of a gas exchange surface?
Well ventilated.
Good blood supply.
Yes, rich blood supply to maintain concentration gradient. And someone said well ventilated, again for the same reason. How many cells thick is the alveoli also?
One cell thick.
Yes, for faster diffusion of gases. Also, it is moist. Don't forget, please study the adaptations of the alveoli and the capillaries and the small intestine. They love adaptations, okay? And the adaptations of the xylem. Any adaptation is important. Now, here we're talking also about the depth of breathing. They show you here the depth here. I wanted to show you that the increase in the height of this wave means there's more depth. And the more the number of waves per second, more frequency. I think we already answered this question. Explain the effect of exercise on the rate and the depth of breathing. We already answered it. Can you say it quickly?
Increases
What happens
rate
to the rate? Yes. In a question like this, okay, shown in figure 3.2 and 3.3. Why did I choose this question? Because if you mention the rate and depth of breathing, it increases, and everything we just said, you will still not take the full mark. Why? You said there's an increase in the rate and the depth to
Explain the
provide
graph.
oxygen. Excuse me?
We have to explain the graph.
Exactly. Thank you so much. If you mention everything we just did, and you said everything, we need oxygen for muscle contraction, for energy, for oxygen depth, everything, everything, and you're perfect, and you did not go back and use numbers from the figures, you lose a mark. So you have to use the figures. You have to calculate how many times the depth increased. Just give any number, at least one or two. They tell you in the mark scheme, quote. You have to quote from the figure. Okay, so you will describe and explain. Good? Okay.
Yes.
Now, excretion. Describe how and where in the body urea is formed. Urea, not urine.
Urea is
How
formed
is
in the
urea?
liver.
Liver.
Very good. And what process will you talk about?
Deamination.
Yes. And what is deamination?
Breakdown.
Separation of the nitrogen containing
Removal of
part
the
of the
nitrogen containing
protein.
part.
Excellent. Yes, the removal of the nitrogen containing part. Now, this is very important. What is Q? It's going towards the kidney.
Artery.
Yes,
Renal
very good.
artery.
Renal artery.
Yes,
Renal artery.
correct. Thank you. Very good. What is P?
Glomerus.
Glomerulus.
Glomerulus. Very good. What is one?
Bowman's capsule.
Bowman.
Capsule.
Yes, the Bowman's capsule. Excellent. What is two?
Loop of Hannah.
This is the loop of Henle, my dear.
Tubule.
Okay, three. This is the proximal tubule. proximal, it's close. And this, in case they ask you, but they
Distal?
don't, the distal tubule, yes, and this is the collecting duct. And all this collectively, it's called the tubules. So this is the Bowman's capsule, and this is the glomerulus, okay? And together called the, you don't have to know, but all this is the tubules. Fine. Now, what is R? That is taking the
Renal
reabsorbed
vein.
material.
Renal vein.
Excellent, lovely. Renal vein, perfect. Okay, these are, of course, microvilli, and they will ask you definitely. Name the structure P. What is P? What is P? What is P? We said it.
Glomerulus.
Glomerulus.
Very good. Okay. And then what do they ask you? Blood vessel Q has the highest blood pressure. Suggest why. Why does Q
To push
have a high?
Blood.
Excuse me? Hmm? Why does
To
Q
maintain a
have a
high
high
So the
pressure
substances can be
for the
forced out
glomerulus.
of the glomerulus.
Yes, you're both correct. This is a branch of the renal artery, if this is an arteriole, or it is the renal artery. And it will give me the glomerulus. And we have to have high pressure in the glomerulus for the reasons you said, for the ultrafiltration. Okay. The structures labeled S contain microvilli. Explain the importance of the microvilli. Now, this is so easy. What is the importance of the microvilli?
Increase surface
Surface
absorption.
area for maximum absorption.
Yes, okay, to increase the surface area, but don't maximum absorption of what?
Fatty acids and glycerol. And
No, don't kill
digested
me.
Water.
No, no, we're not in the intestine. We're in the kidney.
The glucose and water and
Yes. Yes, yes. You just got confused. I think this is Selma, yes? Here, we're talking about the microvilli, my dear, in the kidney, not in the intestine. If we were talking about the intestine, then we will say that those microvilli will absorb the glucose and the amino acids, and the lacteals actually absorb the fatty acid and the glycerol. Okay, Selma? So the fatty acid and the glycerol, my dear, absorbed by the lacteals. Good? The microvilli in the kidney, in the renal tubules, they absorb the, guess what, if you remember, the glucose, urea, what is S? I don't remember. No, it's a W. Water, amino acids. Okay, the soluble, the water soluble materials. Very good. The large proteins are not um, filtered, and the microvilli will absorb what is needed, will reabsorb what is needed. Now, here we're talking about the filtration in the glomerulus. The microvilli will reabsorb what is needed. What is needed?
the glucose and water and
Very good.
salts.
Glucose, amino acids, water, vitamins,
Salts.
minerals. Any urea is reabsorbed?
No, or very little.
No.
Is very.
Yes, no. Urea is not reabsorbed. It is filtered, yes, but it is not reabsorbed. It goes out with the urine. Okay, now, this is more than common. State the name of the growth hormone that helps here. What's the name of the growth hormone in plants?
Auxin.
Auxin. Now, the story. Explain how the growth of the plant shoot is controlled. Now, here, it is growing in the dark. So, what is the stimulus?
Negative
Gravity.
I mean,
Gravity.
phototropism or geotropism?
Geotropism.
Yes. And some of you said negative geotropism because we're talking about the shoot. Correct. Now tell me the story. This is the shoot and it is in the dark. And we said the oxen will make it bend. Where is it? Huh? Where is it produced? How does it go?
Oxygen.
On the tip of
The
the
oxygen in the
At the
tip.
tip. Very
Base of the
good.
tip.
And then,
Used by
moved
diffusion.
by diffusion
correct.
On-
Where will it accumulate?
towards gravity on
At the
the
bottom.
lower side.
Yes. Okay.
Elongate.
And then what will it do?
It will cause some
Um,
elongation upwards.
Stimulate growth.
Very good. Stimulate cell elongation. Yes, which is growth, but please use the term cell elongation. They like it. Okay. And the cells will elongate because they will absorb more water. So that side or A will elongate faster than B. So it will make it bend upwards away from gravity. And this will be negative geotropism. Go revise this part. It's really important. Now, homeostasis. The human body maintains a constant internal temperature. This is an example of what?
Homeostasis.
Homeostasis. And when the temperature moves away from the set point, the mechanism of will
Negative
return the
feedback.
temperature back to negative feedback. Yes. Some students are confused between both. Homeostasis is a definition, which is maintaining a constant internal temperature. How is homeostasis done? The mechanism is the negative feedback. Okay? I chose this so that I clarify this point. Homeostasis is a definition. It is the maintaining of a constant internal temperature or internal environment. Example temperature. How is it done? By the receptors and the brain will. The process is the negative feedback. Okay. Now this. What is K? Skin.
The hair,
The hair shaft. Good. What is J?
muscle,
Yes. What is the name of this muscle?
erector muscle,
Very good. What is O?
nerve endings,
Receptors.
Very good. Nerve endings or receptors. Very good. What's N?
capillaries,
Capillaries.
Excellent. What is M? In case they ask you.
F. Fat.
Yes, fats in the subcutaneous layer. Good. What is L?
sweat glands,
Sweat glands.
Yes, perfect. Okay, we labeled everything. Describe how humans maintain a constant body temperature when the external temperature decreases. And this question is out of 6 marks. Who will remind me? I forgot.
لو عايزين نفتكرها ممكن نرسم الشنت فاسل والكابيلاري diagram ده.
Yes, you can. Of course you can. If you do a small side picture here to remind you, why not?
Sweating
When
decreases.
it's cold, the shunt vessel closes.
Dilate.
1 minute, 1 minute. The shunt will close or open. We're cold, huh?
open,
It's cold.
Open.
Yes.
And the
And we don't
capillaries
use
will
the word
close.
open and close. We use
Dilate and
contract
close.
or
Vasodilation,
dilate.
vasoconstriction.
Yes. Yes.
How
Remember,
does it-
how do we remember? Because because these are several points. Remember the C? I go to the C. Very good. We said, what is the first S?
sweat,
Sweating.
Sweating. Do I sweat or I don't sweat?
no,
No.
Sweat.
What is the second S?
Shunt vessels.
Very good. Do they contract or they dilate?
dilate,
Dilate.
They dilate. Very good. Why?
to reduce heat loss by radiation,
Perfect. So that the blood enters the shunt and moves away from the arterioles and moves away from the capillaries and moves away from the surface of the skin. And the blood will move deeper into the deeper layers of the skin, just like you said, so that I prevent heat loss by radiation or reduce it. I'm trying to trap the heat. Now, what is the last S?
shivering,
Yes. Do I shiver or I don't shiver?
yes,
Yes, we shiver.
Yes. Because when your muscles contract, your skeletal muscles contract, what do they produce?
Heat.
heat,
By
Yes.
respiration.
Now, what is the E?
erector muscle,
Very good. Does it contract or relax?
contracts,
Contract.
moving the hair,
Very good. And when it contracts, huh? What did you say? Yes, the hair will
moves
stand
up,
up or lie down.
stand
Very
up,
good. And when it stands up, what does it trap below it?
air, for insulation,
Air. And this insulates you. But this is not very useful to us. It's useful in other mammals that have
Animals.
fur, like the dogs. Yeah. Okay. And what's A? We actually mentioned it.
arteriole,
Yes. Does it contract or relax?
vasoconstriction,
Vasoconstriction. Perfect. Okay. Now, also, this is a 6 mark question. All this will give you 6 marks, but who will detect that there is a temperature change?
The brain.
Yes, correct. Receptors in the brain and also receptors in your skin, they detect the external temperature and the core temperature inside your body, and they send messages to the brain, and the brain will respond by a motor impulse and will go to your sweat glands and erector muscle and will do the job. Okay, because you can start this question by saying that the temperature change, the stimulus, will be detected by your skin and the brain will take action. And all this we said are the actions. Okay. Now I put here the cold, but also remember the hot. Now this is so important. X, what is X? Synapse. What is X here?
vesicles,
Yes. And what does it contain?
neurotransmitters,
Very good. Okay. Now here they have also an indirect question. They're talking about a disease that you've never heard about called the Parkinson's. It affects neurons in the brain that are responsible for movement. Using the information and the figure, suggest and explain the effect of this disease on a person's movement. You will panic here. You will here look very upset. Why? What will you say? What will you do?
We'll explain how
Lower
the
neurotransmitters in the vesicles
neurotransmitters move.
and lower
Exactly.
diffusion enzyme.
Someone said lower. Lower what? Oh, mean less? Yes. You will look at this figure. This figure is with or for a normal person or a sick person.
Normal person.
Figure
Normal
3.1. Yes, and this is what we studied. We studied the movement of nerve impulses from one nerve to the next across a synapse. This is the normal mechanism that we studied. And this is what you will explain. So in figure 3.1, this is the person that is not sick. And you will explain what is happening when an impulse will arrive. Exactly the sequence of events. Normally, the impulse will arrive, activate the vesicles, it will move towards the cell membrane, fuse. Maybe you're giving all the information provided, very good. And then this is a person with the disease. What do you see? The normal mechanism will be affected. Everything will,
Slow down.
Decrease.
exactly, everything will be less. I have less vesicles, less neurotransmitter, less transmission of impulses. So less muscle contraction. Okay, so again, this is the answer here is the same answer. What happens at a synapse, or what happens when the impulse reaches a synapse? The same answer. I'm going to discuss what normally happens at a synapse or how the impulses will travel. And I will say that in figure 3.2, I am sick, God forbid, everything will decrease. But you cannot just write everything will decrease. What is everything? You understand? That's why I'm telling you, you will state what happens in normal situations, and then you will say that this normal condition will be affected and everything will decrease. Don't just give me, because I think this answer was in a quiz or in somewhere, and most of you wrote that it will decrease. What is it? The number of vesicles, the neurotransmitters, what they do, all the process will be affected. So you have to mention the normal process and say that this normal process will be affected and everything will decrease. Clear? So please, they are smart. You will outsmart them. They are not giving you direct questions, but you will outsmart them and you will know exactly what they want. Clear? In the MARC scheme, they will tell you less vesicles, less neurotransmitters, less impulses. This means that you have to explain what happens normally, and then you will say that everything will slow down or everything will decrease, or at least in summary. Are you with me? What are the two hormones that affect the concentration of glycogen in the liver?
Insulin and
glucose.
glucagon.
Okay, now here is a good chance to remember together. What does the insulin do?
Decrease the glucose level.
Very good. How does it do that?
Store the glucose in the liver.
Very good. As
It as fats.
glycogen, yes. And someone said fats, correct. When I have no more space in the liver, this glucose will be stored as fats. And also, before I even try this mechanism, I will try to increase the rate of uptake of glucose so that I use it in respiration. Remind me, what is the opposite hormone? What is the opposite of insulin?
Glucagon.
Glucagon. What does glucagon do?
Produce
Increases.
Blood sugar levels.
Yes, you're all correct. It will convert the glycogen back to glucose to increase glucose levels. What else hormone also increases glucose levels?
adrenaline.
Adrenaline.
Yes, adrenaline when you're scared, when you have a biology exam. Good. Explain how bacteria become resistant to antibiotics. The story.
Mutation.
Very good. We start with mutation. And then because of the mutation, what do we have?
Very
Yes,
Taking
we
it
have.
Variation. Competition.
Very good. Very good. I have competition between the resistant and the non-resistant strains. And then who will die? Who will survive?
bacteria
non-resistant
dies,
Yes, the
and
non-resistant
the
will die.
resistant
That's what
bacteria
they
reproduces.
multiply.
Very good. You're all correct. Reproduce, multiply. The favorable genes will move on. And this is an example of what? Don't forget the last sentence.
natural selection,
Natural
evolution.
selection.
Yes, thank you so much. That will cause evolution. Perfect. This question is common. It's easy. Now we already answered this question. before. Now the antibiotic sensitivity test. I think I made you a extra sheet. Now this is common in paper 4 and paper 6. Now this part, what does it mean? That we have bacteria or they're dead?
No, clear areas of bacteria.
No, dead.
Yes, they're dead. No, the clear areas, the bacteria is dead. Look, my dear. All this gray part is the bacteria. This is bacteria, all this bacteria, okay? I have here, I started with a Petri dish that is filled with bacteria. I will put different antibiotics. These antibiotics, they will diffuse out. As they diffuse, they will kill the bacteria and they will create a clear zone. So the bigger the clear zone, the more the dead bacteria. Now tell me, choose which antibiotic is the best one to choose for this infection?
E.
E. Okay, I just wanted to make sure that you understand it. Yes. Okay, use the information. Explain why antibiotic E will be the most effective because we just said it has the biggest clear zone. Very good. Or the widest diameter or whatever. Just mention clear zone. How do you minimize the risk of antibiotic B developing the same results as antibiotic A? You will go to antibiotic A. What is wrong with A? Is
Antibiotic
A weak? Yes, it's not effective in killing bacteria. It's not good. So we want to avoid reaching this stage. What do you do to prevent reaching A?
Take full dose.
Take the full
Take
dose,
full
take it
dose.
when you need it.
Take for bacterial infection only.
Not for viral infection.
Very good. Yes, and it's only 1 mark, so this is enough. Good. Now, reproduction, plant and animal. D hormones. What is the role of the FSH?
For the
Stimulate
development and growth of the follicle.
Correct. This is 1 mark. What if it's 2 marks? What else will you write?
Stimuli, LH.
It's produced from the pituitary gland.
inhibits
Okay.
Inhibits
progesterone.
Talk to me
progesterone and
about the
estrogen.
follicle itself. As the follicle itself is growing, what
The
does the
estrogen
follicle
levels
produce?
increase.
Yes.
Estrogen.
Yes, correct. Okay, because this question was in your mock exam and it was out of 2 marks. You all took 1 out of 2 because you all said that the FSH increases the growth of the follicle, which is correct, but they wanted another piece of information. Only very few of you remember to write that as the follicle itself is growing, it will increase the production of the estrogen. And why is the estrogen increase important here? What does it tell the brain? When the estrogen reaches peak levels, what does this tell the brain?
To produce the LH.
Excellent. So these hormones, they are regulating each other. The FSH is increasing. So the follicle itself grows and grows and grows. As the follicle is growing, it is producing estrogen and more estrogen and more estrogen until the estrogen levels are very high. This tells the brain, I'm ready. So the brain will respond by releasing LH, just like your friend said, and this will lead to ovulation. So we mentioned all the 3 hormones now in a minute. There is a 4th hormone that is released after ovulation. What is the name of this hormone?
Progesterone.
Excellent. And what does this progesterone do?
Maintains lining of uterus.
Thank you so much. Very good. Now, if there is pregnancy, who will produce the progesterone?
The placenta.
The placenta. That is in the late stages of pregnancy, correct. In the early stages, who produces the progesterone? The yellow body. Amazing. Amazing. Lovely. Okay, very good. So we revised the hormones here. Now, this common question, advantages and disadvantages of sexual reproduction. Any type of sexual reproduction, what are the advantages?
Variation.
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 Adaptation.
Very good.
Disease.
Yes. I think you memorized it. We said it so many times. Variation, and you have biodiversity and variation, and you're adapted and you can survive and you will not die from a disease and you less chance to become extinct. You show mutation, you allow natural selection, and you will list those advantages in any form of sexual reproduction. Cross-pollination, sexual reproduction, any type, okay? Meiosis, which is, of course, sexual. Now, what about the disadvantages?
More competition.
Needs two parents.
Needs a mate.
You need a
Slow
mate.
process.
Yes. What else?
Connection, isolation.
Isolation.
Very good. You cannot do this alone. You need a partner, you need more energy, you need more time. And let's revise the advantages of asexual. Let's say asexual have advantages. What are they?
Fast only needs one mate, one
Fast
parent.
process,
Very good.
can reproduce
Very
in
good. It's
isolation.
faster. Yes, yes, yes. Perfect. Now, asexual reproduction, this list, do I write it also with the cross-pollination or self-pollination?
Self.
Yes, self-pollination. Okay, they're both sexual, but self-pollination produces less variation because there is less combination of genes. I'm getting my genes from the same flower. Okay, now good. Meiosis and mitosis, very important, of course. Now let's see. Meiosis here, mitosis is a type of nuclear division in which produces genetically identical cells. It is important for what of tissues?
Repair.
Excellent. We repair tissues. We do not repair cells. We replace cells. Okay, so don't forget, we replace cells because when a cell is defective, I will not repair it. It's going to die and I will replace it with another cell by mitosis, an identical cell. So we replace cells and repair tissues. And which type of reproduction needs mitosis? Excuse me?
Asexual.
Yes,
Asexual.
of course, asexual. Good. Just before mitosis, the chromosomes are replicated, and then the chromosomes, what? So that their number is maintained.
Restored.
Double.
No, read the sentence. Just before mitosis, the chromosomes are replicated, so they're duplicated before mitosis. And then
Have a
they are
number.
90, they what?
They divide by two. Yeah, half a number.
Yes, yes, they divide or they split or they separate. Okay? Whatever. Now, I want to exploit the chance and explain this part. The restoring the chromosome number. We have two types or two processes. We have mitosis and we have meiosis. In both scenarios, the chromosome number will remain 46. And the answers are different. How does the chromosome number remain 46? The question that we just answered, mitosis, in mitosis, the chromosome number will remain 46. Why? Because before mitosis, the chromosome number is doubled. Let's say I had a cell with 46 chromosomes. These chromosomes will make a copy of themselves. They will become eventually 90. What's double 46? 92, or I'm wrong?
Yes,
Yeah.
92.
92.
Okay, 92. And then these chromosomes will separate to give me genetically identical cells. They would separate. The 92 chromosomes will separate to give me 46 and 46. So I am back to 46 and I am genetically identical to the parent cell. Meiosis here, we start by talking about the gametes. The gamete, let's say the egg. The number of chromosomes is what? Haploid or diploid?
22. Haploid.
Haploid. I have haploid because the question always asks you in this format, why are the gametes haploid, or what is the significance of having haploid gametes? Because you have a haploid egg that will fuse with a haploid
Sperm.
sperm. Okay. Haploid is a cell containing one set of unpaired chromosomes. Please use this definition. When they fuse in fertilization, what do they give me?
A diploid cell.
Very good. A diploid zygote. That is why also in meiosis, the chromosome number is restored. So the answers are different. In mitosis, why is the chromosome number restored? Because the number of chromosomes before mitosis duplicate and then they separate. In meiosis, we start off with, yes, we do start up with diploid cells and we end with haploid cells. But when these haploid cells fuse together, why are they haploid? When they fuse together, I will have a diploid zygote. Always the question asks you in this format, why are gametes haploid? Or what is the significance about them being haploid? So I hope you understand the difference. This question is very common. Meiosis is another type of nuclear division that is involved in the production of gametes. The chromosome number is halved. From what to what?
Diploid to
Diploid
haploid.
to
Very
haploid.
good. From diploid to haploid to produce genetically different cells, and the fusion of the nuclei by meiosis will form a
Zygote.
Diploid.
Very good. A zygote or a diploid zygote. And this process is known as
Fertilization.
fertilization. Okay. State two examples of how mutation rates can be increased. What are the things that causes mutation?
Radiation and chemicals.
Radiation.
Very good. What if the question is different? They're asking you, what are the things that cause variation in the first place?
Random
Causes.
random mating, fertilization,
Very good. Mutation,
mutation, and
meiosis, random fertilization, random mating. Yes. Good. Describe the benefits of breastfeeding a baby for the first 6 months of life.
provides
Ready
passive
made.
Right, passive immunity.
Yes, yes, yes. This is the idea. Passive immunity, ready-made antibodies from the mother. And this is short-term or long-term?
short.
Short term.
Short-term, yes, but it provides immediate action. Good. Now, in case they ask you, suggest other advantages of breastfeeding, suggest, what would you think of?
Nutrients.
It makes them closer, the baby and the mother.
Yes, it does. And it is cheap and it is bacteria free and it does not cause any allergic reactions and it will make a bond between the baby and the mother and it is for free and whatever. State two ways other than breastfeeding that a baby can acquire immunity.
Vaccine. Vaccine.
Yes, of course, vaccine. What else? Mm. Natural infection, you will acquire immunity through the placenta, through the breastfeeding. Don't forget the types of immunity we have. What we have? Artificial.
Active and passive.
And we have
Artificial.
passive. Yes, we have in both cases natural and we have active and passive and we have natural and artificial in both of them. Okay, don't forget immunity. Now, what is A? What is A?
Stigma.
Stigma.
Stigma. What does it do?
It's where the pollen.
Receives pollen.
Yes, it traps pollen. What is B?
Petals.
That's it.
Petals. What does it do? Okay, it attracts
Attracts
insects.
insects.
What is F?
Sepals.
Yes, and what does it do?
Protects the body.
Protects the baby
Yes, it
flower.
protects the, yes, correct. It protects the baby flower. What does E contain?
Ovule.
It's the ovule and it contains, what is inside it?
Ova.
Zygote.
No, it
Gamete.
doesn't contain the zygote. It contains the female gamete. Okay, the female nuclei. The zygote is diploid, it's still haploid. Now, describe the stages in the reproduction of a flowering plant from self-pollination to fertilization. Now, this is nearly half the lesson. How will you start this question?
Anther produces pollen.
Okay,
And,
look, how you will start, you will define what?
self-pollination.
Yes, start by defining it because this is a good introduction or a good start way. What is self-pollination?
Transfer of pollen from anther to stigma on the same flower.
Very good. Or on a different flower on the same
Plant.
plant. Now, when the anther reaches the stigma, what happens from there?
Pollen
Describe
tube
the story.
formed.
Yes. Okay. So we have here the anther will reach the pollen will reach the stigma, and then what happens?
Pollen
By
tube is
self-pollination, it will germinate into a pollen tube. And how does it move down or how does it create the tube down the style?
It grows down them.
It grows downwards, yes, but it creates this pathway by releasing digestive enzymes that will create a pathway for it to create the pollen tube. So the pollen grain here will germinate into a pollen tube. This is a pollen tube. And what will move down the pollen tube?
the male gamete.
Yes, the male nuclei, correct. You're right. The male nuclei. And then where does it go?
To the ovary and fertilizes the ovule.
Very good. And it enters through a small hole. What's the name of this hole?
Micropene.
Micropyle. Now we have, huh? When they fuse,
They fertilize
continue.
the
Fertilization. Be generous in your answer. Write the whole story. You know what? You can start by a small drawing to remind you. It's okay. The pollen, it will germinate into a tube. It will release enzymes to create a pathway for itself. The main nucleus will move down. It will enter the ovule through micropyle, fusion, fertilization. When fertilization happens and fusion of the haploid gametes, what will this give us?
The zygote, that is the seed.
Very good. That is haploid or diploid?
Diploid.
Diploid.
Very good. So mention the whole story. Now, inheritance. Fine. Okay, inheritance, we study it by solving and solving and solving. This is a sex color disease, color blindness, that is caused by a mutation and the gene is located on the X chromosome. Figure 3.3 is the pedigree diagram of a family who has several people who are colorblind. A lot of you still don't like or get confused with pedigree diagrams. These are diagrams that show how a particular disease is inherited in a family. Okay, let's read on. State the evidence from the figure that supports the idea that color blindness is sex-linked. Now let's go to the figure and let's look at the key. Who is sick?
Only the male,
Males.
only the males
Very good.
have color blindness.
Exactly. So it's very obvious from the figure that only the males, it's more common in one gender and not the other. Not a single female.
And no females have color blindness.
Correct. Perfect. Now, using the symbols X and Y for the sex chromosome and A for the dominant allele for this disease and A small for the recessive allele for the gene for color blindness, state the genotype of person 5. Where is person 5? This person. Let's go to the key. She is a female and she's healthy. So she's XX or XY?
XX.
XX. Now she's healthy, but she came from a sick father. So what is her genotype?
Homozygous recessive.
She's not sick, Habibti.
Heterozygous.
She's
Heterozygous.
heterozygous.
Yes.
She has
a
one
carrier?
healthy allele. Yes, she's a carrier. Very good. She has one healthy allele, one dominant, and one recessive. Okay? Because she came from a sick father, so she definitely took the recessive allele from her father. Okay. Uh, where are we? We said that. Use the information in the figure to complete the genetic diagram. Now, genetic diagrams are more than common to show the probability of person 3 and 4 having another child with color blindness. So person 3, the phenotype, it's a female with normal color vision. Person 4 is a male with normal color vision. Let's go and look at person 3. Look at person 3. She's a healthy female, but her child is sick. So this female is homozygous dominant or she's a carrier?
Heterozygous.
Excuse me? I did not hear you.
Heterozygous.
What is the... Yes. So this is a heterozygous female. So she is XX, A capital, A small. And this male is XX or XY?
XY.
XY, E cap.
Very good. A capital on the X. Please leave the Y alone. Don't put any alleles on the Y. Now, what are the gametes here?
XA capital, XA small.
Yes. Okay. X, A capital, and just Y.
Y.
Okay. And then you will do a Punnett square and you will get the probabilities exactly like this. This girl is homozygous dominant. She's healthy. This girl is also, no, she's heterozygous. It's a small a. So this girl is a carrier. This is a healthy boy. And this is a sick boy. So you will write the genotypes. Genotypes means you will write this like this. Okay? You will write the genotype. Phenotype, you will mention if they are sick or not. How many are sick? The probability of it,
1.
1 out of 4. Okay, 1 out of 4. And it's a male.
25%.
Yes, correct. Okay, protein synthesis. Now this doesn't finish. What is A?
The RNA.
Yes, let's answer this question and we can continue tomorrow. This is the messenger RNA. And what is happening? What is this process?
Copying.
Protein synthesis.
Protein synthesis?
Yes, protein synthesis. We are copying the DNA on a messenger. No, what is A? We said, what is A?
It's the
It's
gene.
coding for it's a gene. Yes, thank you. Thank you. This is the messenger RNA. And the messenger RNA will attach to a what? What is C?
Ribosome.
Ribosome.
Very good, a ribosome. And what is B?
Amino acid sequence.
Very good, amino acid sequence. So we already labeled A. We said A is the gene and the C is the ribosome. Now, describe the events that occur during stage 2, which is protein synthesis in general. What is happening in stage 2?
The messenger RNA is spreading across the cytoplasm from the nucleus to the ribosome.
Yes, but this messenger RNA, before it leaves the nucleus and goes to the ribosome, what did it do to the DNA?
It copied the
It
DNA.
copies the DNA.
Yes, don't forget to say that. This is transcription. It will copy the DNA. After it copies whatever it wants, that gene, from the DNA, it will go out and it will go to stage 3. What is stage 3?
Translation.
Translation.
Translation. Good. Now let's answer this question. State what determines the order in which the parts labeled B are assembled. Who will determine B? The order of amino acids in B?
The base sequence.
Yes, the base sequence on the messenger RNA. Every base triplet codes for one amino acid, and it will finally determine the order of amino acids in B. Okay? Now, 1 minute. Yes, we'll solve this question and then we will leave ecology for tomorrow because ecology is very long. Explain how sexual reproduction results in variation that the scientists discovered in the offspring of plants. How does sexual reproduction cause variation?
Random mating.
Spring not identical.
Fertilization.
Excuse me? Random mating causes variation, yes. What in sexual reproduction that causes variation? You will talk about what in sexual reproduction?
The gametes.
Exactly. You will talk about, yes, meiosis, which is nuclear division or reduction division, where the cells will be reduced from diploid to haploid to give me gametes. And these gametes are genetically identical or not genetically identical?
Not genetically
Not
identical.
genetically identical.
Genetically identical. And we have random fertilization. We don't know which gamete will fertilize with which gamete. And these gametes, when they fertilize together, they will give me different zygotes. That's why sexual reproduction causes variation. This is a tricky question. So you talk about gametes and meiosis and genetically different cells and random fertilization and random fusion of gametes. You say this here. Okay? Now, again, this question that I just talked about. The chromosome number of the offspring plant is the same as the chromosome number of the parent plant. Explain how the chromosome number is maintained from one generation to the next. Here, we were talking about meiosis and gametes. Explain how chromosome number is maintained from one generation to the next. Which answer will you give?
As the meiosis.
Yes. In meiosis, or about what about gametes? What will you say about meiosis and gametes? These gametes contain how many number of chromosomes?
Gametes contain haploid. For when they fuse, when they fertilize, they give a diploid number.
Yes, perfect. You can actually give numbers if you're talking about humans. 23 and 23, they'll give you 46, or they are given a number n and they fuse with another n to give me 2n or a diploid zygote. Okay? Now, this is ecology. Thank you so much for today. We'll meet tomorrow again tomorrow I am going to school. I have class AS. Okay, we'll meet at 3. I'll be back and we'll solve ecology. Okay? Any questions? Did it help?
Yes.
Yes.
Yes, miss. Thank
Okay. Now, of course, like you're doing, any question that stops you, send it to me. And of course, I'll answer you. And I'll meet you tomorrow to finish ecology and revise anything else you need. Good? See you.